Acidovorax. General information. Taxonomy of Acidovorax. Family, Chamaminidaceae. Acidovorax, a genus of bacteria, belongs to the family Chamaminidaceae. This family comprises a diverse group of gram-negative bacteria that are often found in various environments, including soil and water. Formerly Pseudomonas. Previously, Acidovorax species were classified under the genus Pseudomonas. However, based on genetic and phenotypic differences, they were reclassified into their own distinct genus, species. Some notable species within the Acidovorax genus include Acidovorax delafieldii, Acidovorax facilis, Acidovorax temperans. Natural habitat. Acidovorax bacteria are commonly found in both water and soil. Their ability to persist for extended periods in moist environments contributes to their widespread distribution. Clinical significance. Although Acidovorax is rarely encountered in clinical and environmental samples, it can still pose a health risk. Infections. These bacteria have been isolated from various clinical sources, indicating their potential to cause infections. Notably, Acidovorax has been found in blood samples from patients with weakened immune systems, sputum samples from individuals with cystic fibrosis, CF. While Acidovorax infections are uncommon, it's important to be aware of their potential, especially in vulnerable populations. Gram stain. Gram stain characteristics of Acidovorax. Acidovorax bacteria are identified as gram-negative when subjected to gram staining. This means they possess a thin peptidoglycan layer in their cell wall, which doesn't retain the crystal violet stain during the gram staining process. Instead, they take up the counterstain, safranine, appearing pink or red under the microscope. Morphologically, Acidovorax cells present as straight or slightly curved rods. These rods are relatively small, measuring 1.5 micrometers in length and 0.5 to 1 micrometers in width. Acidovorax are observed either as solitary cells or arranged in short chains, further aiding in their identification. Culture characteristics. Metabolic and growth characteristics of Acidovorax. Obligate aerobes / non-fermenters. Acidovorax bacteria are obligate aerobes, meaning they require oxygen for their growth and survival. Additionally, they are classified as non-fermenters, NF, as they cannot metabolize carbohydrates through fermentation. Growth on culture media. The growth patterns of Acidovorax on different culture media aid in their identification. Blood agar, BA. Colonies typically appear yellow and do not exhibit hemolysis, the breakdown of red blood cells. However, the species Acidovorax facilis does not produce any pigment. McConkey agar, colonies are pale, indicating their inability to ferment lactose, a key characteristic of non-fermenters. Acidovorax facilis, in particular, fails to grow on this medium. Buffered charcoal yeast extract agar, BCYE. Acidovorax species do not exhibit growth on BCYE agar. This feature further distinguishes them from other bacteria that thrive on this medium. These distinct growth patterns on various culture media contribute to the laboratory identification and differentiation of Acidovorax species from other microorganisms. Characteristics. Acidovorax bacteria are characterized as gram-negative, medium-sized bacilli that thrive in the presence of oxygen, obligate aerobic growth. These bacteria exhibit positive reactions for catalase and oxidase tests while being negative for indole. Notably, Acidovorax demonstrate urease activity and produce hydrogen sulfide, H2S, in TSI agar. Acidovorax species are motile and display resistance to vancomycin but susceptibility to colistin. Additionally, Acidovorax are capable of utilizing citrate as a carbon source, indicated by a positive citrate test. These distinct biochemical and physiological traits collectively contribute to the identification and differentiation of Acidovorax from other bacterial genera. Poem. In realms unseen, where microbes dwell, Acidovorax, a tale to tell. Gram-negative rod, in chains it may lie, a water and soil dweller, beneath the sky. Yellow colonies on agar's embrace, non-fermenting, it leaves its trace. Oxidase positive, a clue we find, to pinpoint its place in bacterial kind. Though seldom encountered, its presence is known, In sputum and blood, its impact is shown. Vancomycin-resistant, colistin its foe, a microbe's story, in verses that flow.